the last speaker of this session is Marco Palombo. His talk is about imaging almost living tissues, the role of organoids.
So thanks a lot to the organizers for the invitation and to the moderators for the kind introduction. So I start saying that I'm not an organoid expert. So my interest in organoids started a few years ago within the context of the topic of this session. So looking for alternative ways to validate our MRI estimates. And in particular, I focus on microstructure imaging. So we would like to estimate cell size, density, and other properties. And so can we use other systems to validate our measurements? And that's how I got interested in organoids, which is something a little bit underexplored in MRI. As it turned out, there are only 2 published papers with MRI in the organoids. And so today's talk, I will start introducing a little bit what organoids are, brain organoids in particular, why I think they are an interesting system for validation, and what has been done so far with the MRI. So we start saying that for those of you that have never heard about brain organoids are actually 3D neural cultures aimed at mimicking the brain formation and development. And so differently from more typical bidimensional cultures, obviously they add the third dimension. And so neurons are able to grow in an environment where they can create connections in 3 dimensions, and also between neurons connection at the synaptic level, which is mimicking the real 3-dimensional structure of the brain a little bit better than the biodimension. Obviously, this is a toy model of the brain, and the current state of the art is still at the toy model level. However, they are widely used in the neurobiology community to investigate different aspects. For example, the interneurons migration. So it is possible to create 2 spheroids, connect them, and study how cells migrate from one to another to create, for example, cortical layers, or to study specific connections between 2 neuronal populations. And so in this case, it is possible to replicate the thalamic projections, again, using this approach of using 2 organoids connected together. So it is very important to understand what actually they mirror of the human brain, of the brain development. So they do not mirror the adult brain. This is an important thing that when I started looking to them, I got ultra excited of, super cool, we can validate a lot of things. Actually, they mimic the early stage of the brain development, which has pros and cons. In my opinion, the pro is it's very challenging to do MRI in pregnancy. However, there is a developing field there. And so for that field of MRI, organoids could be an amazing tool to validate some of the theory regarding what can go wrong in the development of the fetus, of the brain fetus. And so here you see a scale where it goes from not similar to similar, taken from this paper by Lancaster, which is one of the first pioneers designing the protocol of brain organoids. And you can see that they represent up to 9 months with particular focus between the first and the second trimester of the pregnancy. But how long survive these brain organoids, and at which stage I reached this? So this is rather for you to understand if you're planning to use organoids, this is the kind of time scale you need to look at. So we are talking about months of growth in 3D culture to reach organoids that mirror an interesting phase of the brain development, and then they can last up to 2 years. There are some limitations that I will explain later on. Another important aspect is that this picture, the fact that organoids can only mirror the brain development, is just a technical limitation, is a technical challenge. So this is the current state of the current protocols, but the community is targeting the development of organoids up to old age. And in this paper, again, Lancaster outlines what are the challenges that the methodology needs to address in order to be able to push the organoid's ability to mimic the adult brain. So one, for example, is the introduction of oligodendrocytes. And I can say that myelination oligodendrocytes, actually, we reached this in, so this was in 2016. Nowadays, we can add oligodendrocytes, we can have myelination. So this has been partially addressed, for example. Synaptogenesis, this is rather to do with the actual shape of the organoids. So I will explain later a little bit. The organoids cannot, they do not fold in the cortex, for example. So the synaptogenesis, if the shape of the cortex impact the synaptogenesis, then that is one of the limitation, and so on and so forth. So how do experts in organoids build organoids, is starting from pluripotent stem cells from the donor organism. And this can be a human being, or it can be a rat, or it can be a rodent or a monkey. So, but it's of particular interest, obviously, the human cells. And so this means that for your subject of study, even patients, you can obtain pluripotent stem cells and then start growing organoids in a specific environment, which is a complex protocol, quite expensive, and requires dedicated kind of ovens where very low oxygenation is maintained for days and months. This is because the human brain develops with low oxygenation environment. And so it's still unclear how much oxygenation and the CO2 concentration affect the development. However, once you achieve, once you prepare your substrate, which is just the nutrients that feed the initial cells, then you have two avenues you can pursue. One is the unguided differentiation of these pluripotent cells, and the other is the guided. So the difference is with the unguided, you do not control the final product. With the guided, you actually can control the final product. What I mean for this, I mean that the cells will start naturally differentiating into different cell types that then days by days will grow different brain areas all together. So here you see, for example, this is a ventricle where there is free water. And then you have some areas here, there is cortex that has been growth. And then here is hippocampal-like structure. So this particular organoid, because it was uncontrolled, grows a bit of cortex, a bit of hippocampus, and there's a core of ventricle. What I think is interesting and what the community has been pushing for is the guided one, obviously, because then you can focus, I need a model for the cortex. And so here is an example of rose organoid where these loops here are all small cortices where you have neurons organized in different areas. And what you can do, which is pretty cool, once you have the guided differentiation, you can fuse them together. So for instance, you can grow a cortical spheroid, an hippocampal spheroid, and then make them connect together. And you can study the connection between cortex and hippocampus, for example. And this protocol and this paradigm has been successful. As I said, we can grow cortical organoids. Nowadays, we can grow oligo-organoids, which contain oligodendrocytes, and so there is myelin in there, cerebellar organoids. And so these are just examples of, on the top row, you have an healthy organoid. On the bottom row, it's affected by Lewy body disease. So this means the cells were taken from an individual with Lewy body disease, and it developed synuclein accumulation, which is a typical hallmark of dementia and Lewy body disease. This is an example of organoid, where you can see the myelin staining, and you can see the myelin distribution within the organoid, which is extremely exciting because you can control this to some level. And you can control, you can switch on and off the organoids, depending the oligodendrocytes' ability to myelinate and demyelinate. And then this is an example instead of cerebellar organoid, where you have, again, on the right, this is an healthy organoid. And here the same organoid was induced an immunoresponse, and that's why you have calbimine staining all over the place. So you can even trigger immunoresponses in these organoids and see how the cells and the system react. The research has been pushing this even further. One aspect essential is the vasculature. And the problem is the organoids up to 1 year of culture start developing a necrotic core because the nutrients, this cell agglomerate is embedded in the nutrients. And so only the cells at the surface get by osmosis the nutrients, and the core start dying and entering necrosis. To avoid this, we need to vascularize it so that the vasculature brings nutrients inside. And that's the biggest challenge currently. There is not an easy way to build a vasculature within an organoid and make it sustain. Even if you build the vasculature, how do you pump the blood inside and outside? So that's the major challenge. The interesting approach, when I said you can grow a couple of spheroids and bring them together, that's a simplified version of what we can actually do today. Actually, today we can create what is called multilineage assembloid, which is slightly more complicated than just taking two separate brain regions and connect together. So the reason of the higher interest in multi-region assembloid or multilineage assembloid is that they are designed and validated to mimic also the activity of the brain. So these neurons spike and interact between each other. So this is a living organism that you can potentially scan alive in your scanner. In terms of what pathologies they are able to mirror to some extent, we have not only, for instance, microcephaly induced by the Zika virus. This was one of the first striking application of brain organoids. Nowadays, they can, as I showed you before, we have an example of degenerative disease where the organoid go under neurodegeneration. There are organoids that can mirror autism spectrum disorders, abnormal and healthy development, and then we have tumors. For tumors, the community split whatever tumor organoid it is, they call them tumoroids, and it's a separate line of research. I will focus on brain organoids. This is just an example of Alzheimer's disease. In this case, they created an assembloid mirroring the cortex, and then they got two assembloids. In one, they injected microglia-like cells carrying the high-risk allele ApoE4. In the other, they injected microglia-like cell carrying the low-risk ApoE3. And what they demonstrated using this living system is that naturally organoid with the cells carrying ApoE4 were accumulating more beta plaques in their cells, and they were expressing more 88, which are two of the hallmarks of Alzheimer's in the adult brain. So although this is a developing brain model, they showed it can replicate some aspect of disease also in adult. These are just some of the limitations that I already discussed. The major one is the necrotic core, and I will come back to this in a second when I show you the MRI. So you want organoids to be as big as possible, and the scale here is actually not always reachable. So 4 millimeter, yes, you can get it with 4 millimeter, but over 2 millimeter of it is just death tissue inside. So that's one of the big challenge to address. There are several interesting solutions to this, and one involves the vasculature. The one one obvious idea could be to take the organoid, implant in an host, and let the host grow the vasculature. And that's done very often, actually. The organoid is embedded, for instance, in rat or mouse brain, and the rat and mouse brain start growing the vasculature, feed the nutrients, the organoid continue to grow. But this could be, for instance, human cells expressing human disease inside an host organism, which is instead a rodent. And so there are many studies showing, and this is a nice literature review, where they show that the implantation of organoids in external bodies doesn't impact the behavior of the cells within the organoid. So when it comes to MRI, what are why these are? I think these are interesting validation systems. Well, first of all, we can control quite accurately the cell populations. And so if you are interested in targeting imaging by MRI biomarker of microglia, for instance, you can target that cell population and manipulate the presence in the organoid of that cell population. You can control the chemical environment, which means you can create organoids that technically we saw from the talk before could contain more of one kind of ion with respect to another. And you can test within a living system how this get uptaken by the cells, where it goes and what it happens, for example. We can study different level of migration. In particular for cortex, you can develop organoids that can express different layer structure. So they are not really capable to have all the 6 layers of the human cortex, but you can play up to 3 layers, and the connectivity across layers can be tuned. And then it's a live object, it's a live being. So which means, as I said, the neurons fire. And so you can start perhaps investigating also the functional activity of this very, very toy model, very simplified networks of neurons. But can we actually measure them with MRI? And so here for the remaining, I think, 3 minutes, I'll show you the only few applications I found in the literature, because it's a very, very new system with so far didn't receive much attention from our community, in my opinion. So in fact, the very first study using MRI came out recently, and they were not using MRI to actually characterize the organoid per se. They were just interested to find a way to characterize the overall shape of the organoid longitudinally. So people usually section organoids, and this is quite of a limitation for that field. They would like a technique that allow them to scan organoids longitudinally. And so why not MRI? And here you see they put the organoid in a tube. This is the organoid here. And here they developed an AI system to automatically segment within the noise the organoid and follow it, segment different parts of it and follow the different aspects of it. And so the focus of this paper was absolutely not the MRI. It was more using an MRI as an object, as a useful technique to image the organoid. The second one instead was very much targeted at, shall we use MRI to characterize the architecture of the organoid? And so in this work, they actually performed tractography of organoids, and I think it's pretty impressive. Just a note, very high field. Both studies used very high field scanner. And you see here the challenging in scanning the organoid. This organoid was less than 2 millimeter, approximately 2 millimeter in diameter. You have to keep it in the substance, so in this medium, which has its own benefits and cons when you do MRI. And you don't want to fix it in any way, so it can move around. So it's quite challenging to register the organoid and to scan the organoid. In this case, they didn't keep the organoid in the medium. So this organoid died during the scan, but what we did, we kept it in the medium, and you can get pretty impressive connections within this organoid. One cool thing of this study is that this is not an organoid having oligodendrocytes, so there is no myelin here. Yet, doing DTI, you can still reconstruct how the long projecting axons, unmyelinated, are arranged in the space. We ourselves did some experiments. This is unpublished material just before COVID. And then the reason it's unpublished is because then there was COVID and our organoids died. And then, we couldn't continue the research. But here we characterize that more accessible preclinical field, high resolution, and we quantified the T2, the T1, and the diffusion of a couple of organoids. Changing different different echo time, repetition time, and B values and B directions. So what I want to point this out is that this was a relatively big organoid. It was roughly 2 millimeter without necrosis at the center. And you can get very good, very decent images. You can do quantification on this. And so these are the distribution of T1, T2 mean diffusivity, and they mirror quite well the cortex, the distribution in the cortical layer. And here you can see the distribution of the fibers. Again, this organoid didn't have oligodendrocytes, and yet we can recover the organization. This was let's say, the brother, and this organoid had also a little ventricle here with free water, and a different organization of the main cortex, and then you could follow fibers going down in this area with higher fractional anisotropy. What I'm very, very, very excited to see at this meeting, and in particular on Friday, this beautiful work from Julia Krug, which will present actually MRI at ultra high resolution. They used the 28.2 Tesla and they scanned the organoids and they quantified their properties also as a function of time. So I strongly encourage to have a look at this work on Friday. And what they showed is that, which I think is extremely interesting. They showed similar results as the one we obtained in our unpublished data in terms of the diffusion properties and mean diffusivity, but they also did localized spectroscopy. And the localized spectroscopy, they show that the localized spectra indicate metabolite changes. So we were not able to do localized. We tried at 9.4 Tesla. Because we had the organoid embedded in the medium, we were not able to suppress the outer signal. Probably at 28.2 Tesla, this works much better. And they also quantified time dependence of the diffusivity and the kurtosis. So just to conclude, because I'm slightly over time, I think I gave you an overview of how organoids are 3D cultures that can be useful to us to study and validate some of our techniques. MRI can characterize organoids longitudinally and in vivo. And we have examples, although very limited, of applicability of MRI to organoids. However, it requires preclinical scanning at very high field for the moment. Localized spectroscopy seems to be feasible, which open opportunities to study also the metabolism of these interesting systems. And with this, I would like to conclude. Thank you all and our team in Cardiff. Thank you.